we're going to get started. A quick announcement before we get started. There have been a few schedule changes for tomorrow, and I am going to be sending you a PDF of tomorrow's schedule via email, probably within the next hour. So please be on the lookout for that, because that will replace what's in your book for tomorrow. I'd like to introduce Brad Seibel, who's here from University of Rhode Island to talk about physiological effects of ocean acidification.
Thanks. All right, well, thank you guys for inviting me here. I'm not wearing the microphone for the course here, so for the classroom, so let me know if you can't hear me in the back. I can speak up. So you've been hearing about some of the difficulties of measuring pH in seawater, and hearing that, I think it was Andrew Dixon that said that the difficulties in measuring the behavior of organisms is by far and away the more difficult part of this whole procedure, and I think he's correct about that. May not be more difficult procedurally, but it's certainly more difficult to interpret the results that you get and to get results that are consistent. And I'll try to explain why that is and what you can do to sort of head off some of those difficulties. This is an equation called the Henderson-Hasselbalch equation, a quick sort of reference that physiologists have used to predict what should happen to pH bicarbonate as you increase CO2 concentrations, and I'll come back to that. So my lecture today, I wanted to introduce acid-base physiology, exactly what organisms do to correct for changes in the acid-base state of their tissues and their extracellular fluids, namely buffering capacity, what is the ability to resist pH changes as CO2 increases, and ion transport, the ability to pump out proton equivalents or exchange protons for other ions in order to compensate for acidosis. And there's a correlation between the capacity to do those things and the metabolic rate, and I'll explain why that is as I go along. So processes that are sensitive to high CO2 or low pH are limitless, basically. Any enzyme-mediated process is potentially limited by pH, or there's a pH optimum for most enzyme-mediated processes, and so CO2 has the potential to alter those processes. There are a couple, though, that are particularly important, or at least that we are particularly worried about. One of those is blood oxygen binding, and I'll spend the last half of today's lecture covering that in some detail. And I also wanted to look at metabolic rate and whether it's a good indicator of what the effects of CO2 might be, why people tend to measure metabolic rate, and how you interpret that result. So I'll end with a particular case study. Dosidicus gigas is a large squid in the eastern tropical Pacific where there's a very pronounced oxygen minimum zone. So we spent a lot of time studying that animal and how it migrates through a gradient of low oxygen temperature and CO2, and explain, mostly because of its blood oxygen binding characteristics, why it is potentially a canary in the coal mine. And if there's time at the end, I wanted to address this question of whether it's possible to pinpoint a critical CO2 level that we need to avoid for animal physiology. And I'll tell you up front that I think the answer is no. I don't think it's possible to pinpoint a single critical CO2 level. And by way of analogy, physiologists have been studying the effects of low oxygen on organisms for at least a century, and I don't think we could pinpoint a critical low oxygen level. And the same processes that control oxygen transport are often the same processes that control CO2 excretion and the ability to control CO2 levels. And so they're species-specific problems there. And so if you want to try to find a single defined critical level, I think you're asking for trouble. All right, so the most important point is that organisms can control intracellular carbon dioxide partial pressure and pH. So just because the pH changes externally or the CO2 changes externally does not mean that their blood or their intracellular pH and PCO2 will change. These are pictures of hydrothermal vent organisms that live near venting water that is extremely low pH and high PCO2, and they manage to maintain intracellular and extracellular levels that are equivalent to what other organisms in other environments experience. So this is the beginnings of a Davenport diagram that physiologists use to monitor the effects of PCO2 on the acid-base balance within an organism. And I'll walk you through this in steps. But so this is these graphs were taken from a textbook that focuses on human physiology, and so human PCO2 is 40 millimeters mercury in the blood. It's very high, which is another important point that most marine organisms maintain a much lower PCO2 than air-breathing organisms for reasons that I won't get into, but it's just another indication that just because PCO2 is high does not mean that cellular functions can't happen. So what this is showing you, if you think back to that Henderson-Hasselbalch equation I showed you, if PCO2 goes up but you hold pH constant, then bicarbonate must increase. So you end up with a higher bicarbonate for a higher PCO2 level with roughly the same pH. So you'd end up here. Higher bicarbonate, same pH, but a higher PCO2 level. Similarly, if you maintain constant pH but lower the PCO2 level, you'll end up at a lower bicarbonate level. So you can identify lines on this curve that have a constant PCO2 level for given characteristics of bicarbonate and pH, shown here. So anywhere along this line, you have got a PCO2 of 40 millimeters mercury, but changing bicarbonate and changing pH levels. And you can identify several, or an infinite number of lines on here for different PCO2 levels, as I'll show you in a minute. So if you increase PCO2 from 40 to 60, but you hold the pH, or excuse me, you hold bicarbonate relatively constant, then pH drops. And so if you have an animal that you have in a chamber and you increase the PCO2, the pH is going to drop along some characteristic line, and that line is the buffer line. It indicates what capacity the organism has in its cells or in its blood to buffer that pH change, and this happens very quickly. So as soon as you introduce high CO2 to an organism, the pH is going to drop by some amount. That's a species-specific buffering capacity. And again, for each of those new PCO2 levels, you can identify isolines. So anywhere along this curve is a PCO2 of 60 millimeters mercury, 80 millimeters mercury, and in this direction, lower PCO2, 27 and 17. So the slope of this line tells you what the buffering capacity is. If this is steeper, it means you've got better buffering capacity. You get less of a pH change for a change in PCO2. So if you go from 40 to 60 and you get a change from pH 7.4 down to 7.2, if you had a better buffering capacity, the same change in PCO2 might drop you down to 7.3 instead of 7.2. So the steepness of that line tells you whether you have a strong buffering capacity or a weak buffering capacity. So this particular graph is taken from example buffering capacities in different cells within an air-breathing mammal. When you get into the marine environment, or when you start comparing different organisms that are more distantly related, have very different metabolic rates, buffering capacity can be widely varying from one species to the next. So you've probably heard terms like respiratory acidosis and alkalosis if you watch any hospital TV shows. Basically anything that changes the PCO2 but bicarbonate stays relatively constant, that's a respiratory acidosis, because you can change your CO2 level through respiratory processes, breathing in and out. So if you hold your breath, you're increasing your PCO2 internally, and your pH is going to drop. That's a respiratory acidosis. If you hyperventilate, you're driving off CO2, and you'll end up with a respiratory alkalosis. Bicarbonate is not changing. You can think of this as analogous to changes in total alkalinity. So alkalinity is remaining constant while PCO2 and pH are changing, if that helps you think about it. Along a constant PCO2 line, you can get a metabolic acidosis. So processes that control how much bicarbonate you have in your tissues or your blood. If you have a kidney malfunction, you can end up with a metabolic alkalosis or acidosis with the same PCO2 level. So you can then compensate for those imbalances. So if you have a metabolic acidosis, you could bring your pH back toward normal by hyperventilating, driving off more CO2, and you'll come back toward your control pH. A different bicarbonate level, but the same pH. So organisms do this over some period of time that is also species specific and depends on their capacity for ion transport. So this is a typical response to a change in PCO2 for any organism, any marine organism. You'll get a response immediately within a few minutes of changing the PCO2. That's dependent on the buffering capacity. And then over the next hours to days, you'll get some compensation for that through ion transport processes. You'll increase your bicarbonate levels and return toward the control pH. So there's some return toward control pH, but you'll notice it doesn't go all the way in this case. That's very common that you'll get a partial compensation rather than complete compensation. So I mentioned that the capacity to control intracellular pH is an evolved function that depends on the rate of production of acid-base equivalents. So during regular, normal cellular metabolism, mitochondria are producing protons, you're producing carbon dioxide, and normally carbon dioxide is excreted. You've got buffering going on, you've got ion transport going on all the time. And if you have a high metabolic rate, you're producing more CO2, more acid equivalents, and you've got capacity then evolved to get rid of those CO2 and proton equivalents. If you have a low metabolic rate, you're not producing very much CO2. You don't need such high ion transport capacity or such high buffering capacity. So you may not have that capacity inherent. And so in this paper that came out in 2001, we were predicting that organisms with low metabolic rates would be more sensitive to high CO2. And this was in the context of ocean carbon sequestration. So if you take carbon dioxide before it goes in the atmosphere and pump it purposely into the deep sea, you could keep it out of the atmosphere. And it was argued by some that there aren't many animals in the deep sea and that there would be no effect of this. But deep sea animals tend to have lower metabolic rates, both due to temperature and evolved low locomotory capacity. So we were predicting that they would actually be more sensitive than some shallow living organisms. So it's a broad general guideline that's got some support since then, but remains to be tested whether this can be a general predictive guideline for what organisms are going to be most sensitive remains to be seen. So this is from a recent paper by Melsner et al. In Biogeosciences, where they show a response of three characteristic organisms, one that has very little ion transport capacity and low buffering capacity. And you can see that for a change in PCO2 from 0.13 to 0.53, you get a very large drop in extracellular pH compared to these other two organisms that have slightly better buffering capacity. Get a smaller drop, and then over time recover toward the control pH. I think they were hypothetical, but I'd have to go back to the paper and look, actually. I don't know offhand. So when I say that there's a lot of variation in metabolic rate, this is just to give you some idea. These data are all cephalopods, squids and octopuses that I've measured over the years. Shallow living, very active squids to deep sea vampire squids that are very gelatinous and soft-bodied. And this is a metabolic rate on the y-axis, and that's a log scale. So there's more than 200-fold difference in metabolic rate between a vampire squid and a shallow living squid. And similarly, you get that large of a difference in buffering capacity as well. and ion transport capacity. All right. So just to walk through these steps in a little bit more detail and show some data, this is just representative of buffering capacity. You've got some species represented here by A- that can soak up protons. If you add CO2 to the cell, you get protons produced. Those combined with some buffering species and produce this compound. And so you're effectively removing protons from the intracellular space. And this is correlation between metabolic rate and buffering capacity in those same organisms I just showed you, the cephalopods. So you get buffering capacity of 100 slikes in some of these shallow ones, and a slike is named after the guy who first measured buffering capacity in the early 1900s. And the way you do this is simply by titrating a homogenate of muscle tissue with acid, and it's defined as the micromoles of acid required to change the pH from 6 to 7. So it's a very simple, straightforward measurement, and you get huge differences in the amount of acid required between a deep and a shallow living, or a fast and a slow organism. Yep. So this is high metabolic rate over here.
Is there some reason you do it that way?
Yeah, actually this graph was originally made, so this is actually versus habitat depth. But I thought for habitat depth correlates perfectly with the oxygen consumption rate, and so I just substituted. So it's not actually metabolic rate, but it's a very close indicator of it.
Question for you. Wouldn't you expect that, do you correct to the fact that when you take these deep sea organisms up, there must be an effect on the buffering capacity because of pressure change? How do you correct for the...
There's a long and sordid history of looking at pressure effects in deep sea organisms. All of these organisms were captured in the top 1,000 meters, and all of them can be maintained alive on board ship for weeks. And so pressure doesn't seem to have a strong effect on their overall physiological functioning.
I'm wondering about just the basic chemistry. If you're measuring, if the measurement's made by the titration of, of hydrogen photons,
Mm-hmm.
measuring the buffering capacity of effectively a solution, it would seem that that's got to be a function of pressure. When you change the pressure,
It's possible that there's some effect there. We're measuring them all at ambient pressure. If you take these down, I mean, there's many different correlates that have been measured to get around this pressure question. So you can look at the concentration of mitochondria, you can look at various enzymes, ion transport proteins, you can measure total water content. They're basically, these deeper living ones are basically gelatinous goop. So if there's some pressure effect, I would be very surprised if it would make up for this 100-fold variation. But I don't know anything about how pressure affects buffering in particular. But I'm 100% confident that the pressure difference doesn't account for the difference in metabolic rate. I mean, you can pick the animals up and see that there's no way that this vampire squid has a metabolic rate anywhere close to a surface squid. It just runs through your fingers. There's nothing to it. So this is data from a deep and a shallow living squid, just showing buffering capacity and what the effect actually has on the pH. So you get a much larger change in pH for an equivalent change in PCO2 for a deep versus a shallow living organism. So ion transport, as I mentioned, there are a number of different mechanisms. You can pump protons directly out with proton ATPases, which of course requires energy. There are exchanges with cations, other cations. You can pump bicarbonate in, you can pump chloride out. So there's a number of different ways of going about that. But the net effect is that over some period of time, usually within 24 hours, you've returned to some steady state, new steady state level, and we'll maintain that. And this new steady state seems to be a trade-off between ionic balance and acid-base balance. You rarely see intracellular pH you rarely see extracellular pH come all the way back to the control level. There also seems to be some indication that extracellular pH is sacrificed for the benefit of intracellular pH. Intracellular pH is better compensated than extracellular pH generally. So this is some data on a variety of organisms, including bivalves, worms, and crustaceans. On the left, and this is a log axis of carbonic anhydrase, and the units dropped out for reasons that are unclear. But these are organisms from hydrothermal vents that experience very high CO2 levels, very low pH, and they have very high capacity to deal with those conditions. They can carbonic anhydrase is an enzyme that catalyzes the conversion of CO2 and water to bicarbonate and protons. So it's very important. You find high concentrations in gills, intracellularly, in stomach lining where you're secreting a lot of acid. Any place where you've got any sort of acid-base adjustment going on in an organism, you'll find carbonic anhydrase. And organisms at the vents tend to have very high levels compared to even shallow living species that are producing a lot of CO2. They have high metabolic rates, but they don't require the same kind of capacity to deal with changes in their environment like the vent animals do. And then deep living non-vent species tend to have very low levels because they experience only very small changes in CO2 in their environment. So there's huge literature on measuring intracellular pH, including this book that came out in 1989. Since then, methods, I can go back to that if anybody wants to write it down, but modified by Portner et al. In respiration physiology, they determined intracellular pH and PCO2 after metabolic inhibition by fluoride and nitrile acetic acid. And the idea there is that if you have a homogenate of tissue, that there's still metabolic activity going on, and it's changing your PCO2 and your pH while you're making the measurement. And so this is just a modification of earlier methods to inhibit that, so you can make a more accurate measurement. And it was recently validified in fish, looking at tissue that was stored for long periods of time versus others that weren't, and found that the method was quite reliable. There are also methods, some more recent, using optical techniques, using dyes, pH-sensitive dyes that you can actually get inside of a cell, so you can look at live cells under a microscope. And there's some more recent work looking at transparent animals, so larvae and zooplankton that are transparent enough and small enough you can get some of these pH-sensitive dyes inside a whole living animal and measure changes in intracellular pH. And of course, your time course of these measurements is much better. Over just a few minutes, you can see these changes happen. But it takes a certain type of organism or isolated cells to really be effective at that. And the last major method that organisms have to protect acid-base status within the blood and in the tissues is through transport in the blood. You can transport protons, you can transport CO2 directly. So any protons that are produced in the cell are bound by, in this case, hemocyanin. HC is hemocyanin, which is a respiratory protein found in crabs that you'll hopefully measure pH in later today in the lab. Or hemoglobin, which is what fishes have. And that proton, as it binds to the hemocyanin, reduces the affinity of hemocyanin for oxygen. And so oxygen is kicked off at the tissues where it's needed. And so this is an adaptive mechanism to enhance oxygen offloading at the tissues. So if you have a system where the blood is especially sensitive to these protons to enhance oxygen transport, and I'll argue this is the case in the cephalopods, the squids that I'm coming to later, if you have an especially sensitive respiratory protein to pH, then it's going to have trouble binding oxygen in the first place at the gills. It'll still be able to release whatever it binds at the tissues, but it's going to have a hard time picking up oxygen in the first place. So there's a number of questions. You can show that organisms can compensate, at least partially, for changes in intracellular or extracellular pH, but it's not clear over a long term what that means for the organism. There have not been any, or at least relatively few, well-controlled studies of acid-base balance under high CO2 in organisms. So it might be that you can tolerate very high bicarbonate levels as a compensatory mechanism to protect your acid-base balance for days, weeks, or even months, but over some time period, that imbalance in the ionic status will have its effect on some other process. It may be that it takes a lot of energy to maintain that balance in terms of ion transport. So that's a very wide open question in my mind, exactly how long can an organism maintain a compensated acid-base balance. And whether there is an actual energetic cost and whether that's measurable is another wide open question, I think. There clearly is an energetic cost at some level, but how important it is and how much greater is that cost than normal acid-base balance is still unclear, and whether it's possible to sort that cost out from the multitude of other processes going on. It's very difficult. And the last thing that's a wide open question in my mind, and where people are starting to get a handle on this, as you'll hear tomorrow in tomorrow's lecture, that organisms can produce new isozymes or they can upregulate pathways to produce more of existing enzymes. And so there's some ability to acclimate that may also have its costs or benefits. But the capacity for acclimation is largely unknown. Add to that the capacity for evolution, and the time scale required for evolution is a whole other question that we may never answer. All right. So I mentioned enzyme-mediated processes have an evolved pH optimum. This is just showing an example of pepsin from the stomach has its optimum function at pH 2, where you would expect that's the pH of your stomach acid. And so pepsin, which is important for breaking down proteins, is optimum at that pH compared to trypsin, which works at much higher pH. So there's some capacity for creating new enzymes that have different pH optima, and it's unknown over what time scale that can happen or what capacity organisms have inherently to do that. So there are qualitative as well as quantitative changes that you can make in enzyme capacity in response to high CO2. The whole organism level is a much trickier bag. So this is a sort of energy budget. You've got ingested chemical energy. Some of that is absorbed and used for biosynthesis, maintenance metabolism, generation of external work, muscle contraction. And some of that energy is shunted toward growth. Some of it's lost as excretion and is measurable as heat. So all of these processes are interrelated, and if you measure a metabolic rate or a growth rate, you're actually measuring the sum of many, many different processes that may all have different pH sensitivities and CO2 sensitivities. And it's very difficult to tease those apart and figure out exactly what has changed and how to interpret that. Another thing to keep in mind about metabolic rate is that it represents a cost to organisms. You'll sometimes see, such as this quotation, a simplistic idea of metabolism as a benefit to an organism. If you have a higher metabolic rate, that's necessarily a good thing. But because metabolic rate is the sum of the costs of all the processes, of all the work the organism is performing, a high metabolic rate means higher cost. It's not an advantage. You may require higher metabolic rate to perform the work that you need to do for predator-prey interactions in your particular environment, for example, but it's not an increase in fitness to maximize metabolic rate. So this is just a pie graph I threw together. These are not real numbers, but just to give you some indication that going from a control to a treatment CO2 condition, you can change any number of things. It might be that the animal is stressed out and is trying to escape from your chamber more, so you get more muscle contraction, more activity. It might mean you've got a reduction in synthesis macromolecules might mean that your specific dynamic action has decreased. This is the cost of digestion and fuel. Maybe that ion transport is decreased, but you won't see it in measuring a whole animal metabolic rate because of this change in activity. So you've got the same metabolic rate in these cases, but things have definitely changed. And in a short-term experiment, it's really difficult to figure out exactly what has changed and what you've measured. So, how do you measure a metabolic rate to increase your chances of measuring something meaningful? There are a number of steps you can take, number of things you need to control. The duration of the experiment is 1. So if you have organisms that are stressed out, they're going to have an artificially high metabolic rate. The metabolic rate of a stressed organism can be tenfold higher than an organism that is relaxed and adjusted and acclimated to its chamber. If you're measuring a stressed out animal, you have no chance of figuring out what has changed due to your treatment. You have to maintain oxygen well above the critical partial pressure of oxygen, which again is species specific, so you have to determine that ahead of time. You have to know at what point oxygen consumption rate starts to fall off, at what critical partial pressure of oxygen, so that you can maintain the PO2 above that. Typically, if you maintain seawater above 50% saturation, you're safe, but that's not a hard and fast rule. The respiratory quotient is the ratio of CO2 excreted to oxygen consumed. So if you are enclosing an animal in a chamber and allowing it to consume oxygen, you're also allowing it to produce CO2. And depending on what fuel it's burning, that amount of CO2 will vary. If you're burning purely carbohydrates, the ratio of CO2 to oxygen is about 1. If you're burning purely protein, it might be as low as 0.7. So that CO2 is going to change your pH inside your chambers. So you have to take that into account. So if you're maintaining your oxygen at a high level, you're also maintaining your CO2 at a relatively low level. But even so, you still need to be able to measure it and know what it is. You have to maintain temperature within the habitat range. If you measure outside its normal range, you're going to get a stressed out animal. See 0.1. Q10 is the measure of a change in activity, change in a metabolic rate for a 10-degree change in temperature. And it's usually in the range of 2 to 3, meaning you'll get a doubling or a tripling of a metabolic rate for a 10-degree increase in temperature. If you get outside the normal temperature range, that Q10 can jump up to 5 or 6 or 7 or who knows what. You have to control activity level, aerobic scope, going from a resting metabolic rate up to an active metabolic rate can be 5 or 10-fold. So it's very important to control for activity level. Physiologists have defined a number of different metabolic states. Basal is considered the gold standard, and it's only achievable in mammals, and that's even questionable, where you can have a mammal in a chamber, you know that it's resting, you know what it's eaten, you know what its body temperature is, and you can control for all sorts of variables that you can't control in an actively swimming squid, for example. So most marine physiologists talk about routine metabolism. You've done what you can to control for these things, but you put it in a chamber in the dark and hope for the best. It does what it does, and that's the metabolic rate you get. If you're actively controlling locomotion, you can make it swim faster or slower, and then you can set your desired activity level. That's workable for some more active fishes. It's very difficult to do in slugs, for example. You can put crabs actually on a treadmill. There's video of this on YouTube. Not kidding, I'm not making that up. You can also control feeding. The specific dynamic action, as I mentioned, can drive up metabolic rate. So if an organism is eaten recently, for the next several hours, its metabolic rate could be 2 to 5 times higher than the basal metabolic rate. Lastly, chamber volume and flow rate are critical. So if you're doing a flow-through sort of experiment, and I'll talk about that in just a second, the rate of flow through the chamber is critical, as is the volume. If you can control these two things at an adequate level, then you're simultaneously maintaining oxygen and CO2 where you want it, and you're not stressing the animal. out.
Can I ask a naive question before you go on?
Please do.
So thinking about some organism like a larval coral or a larval mollusk or a sitting mollusk.
Yes.
How do you think about activity levels for somebody like that?
Routine. Some microorganisms do have quantifiable activity levels. Like there are pelagic animals that swim more or less continuously, but maybe at different rates. I've seen setups where you can put a little shrimp attached to a wire that gauges how fast it's swimming or not. You can videotape them and look at, like in jellyfish, the rate of bell contraction. So you can quantify locomotion in some of these things, but it's going to be, again, species specific, and it's going to take some playing around. There aren't any methods worked out for most organisms. It's a good question. Oops, not too far. So this is just an example of some data that we collected in our lab for small squids. And what this is showing is that depending on whether they were in a 300 milliliter BOD bottle or a 50 milliliter syringe, you got very different answers. For one, they had much higher metabolic rates than we anticipated they would, and so the oxygen at the end of the experiment dropped down to less than 20 micromoles in some cases, and that makes up a lot of this difference you see between the two treatments. But there also seems to be an effect, even here where the ending oxygen concentration was fine, you still see, I'm not sure I'm reading that right now, but there's some indication that just being in a smaller chamber stressed them out more, and they shut down. Rather than having a higher activity level in a stressful situation, they seem to just clamp down and stop swimming around as much. So you had very different metabolic rates depending on whether you confine them to a smaller chamber or let them have some room to swim around. And it becomes very difficult to interpret which is the case if they're swimming around more or if they're shutting down. You just don't really know. And so the best you can do is make sure that your treatments are all the same and that they're not reaching a critical oxygen level. So this is not acceptable. You have to avoid that, otherwise it becomes impossible to know what you've measured. These were experiments that were what we call endpoint, which is the way you would do a lot of these larval measurements as well, or at least that's how I've seen it done in some cases. I saw one, getting back to larvae for just a minute, talked with Adam Marsh, who's at the University of Delaware, and he does a lot of larval physiology, and has come up with some very ingenious ways to measure some of these things, actually. He puts larvae in individual well plates on a plate reader, and he's got optical stuff on the bottom that determines the oxygen level optically, and he can put a whole plate in with 196 larvae and measure their oxygen consumption rate all simultaneously. So it's pretty slick. But he told me at one point he'd developed a system to measure it with an oxygen electrode, and he was convinced that the electrode itself, turning it on or off, would depress the metabolic rate of the organism. There's some electrical activity, I don't know. So that's a challenge. But what he ended up doing was creating little micro BOD bottles of about 500 microliters and incubating them for some period of time, taking a water sample out and injecting it past an electrode. So that seems to work. But as an endpoint measurement, you don't have any control over where you end up. It's guesswork as to how much oxygen you're going to have at the end of the experiment. You have to time it right with the volume and the number of organisms in the chamber, etc. So do you want to use electrodes, or there are what are called optodes, which are optical oxygen sensors. I believe in Portner's chapter in the best practices guide, he leans toward optodes because you reduce the requirement for stirring mostly, and they're less sensitive to temperature. However, you have to stir anyway, and you have to control temperature anyway. So it doesn't really matter in my mind which you're using. And oxygen electrodes, if you buy them off the shelf, they're about equivalent cost. And the ones that I'll have in the lab today were store-bought off the shelf. You can make your own oxygen electrodes for practically nothing. You can make 100 of them for the same cost as buying one off the shelf. and some fraction of them won't work, but you can do that. So if cost is an option, electrodes are a better way to go. Calibration is an issue. You have to know what saturation is. So you can bubble seawater with oxygen, but then there's some concern that you're oversaturating, and you have to control the temperature of the air coming in so that you don't heat up the water. So usually just some gentle stirring and being patient will get you at an air saturation level. And at the other end of the spectrum, you can bubble with nitrogen and drive all the oxygen off, which works, but it also requires patience and setup to control so that you're not allowing any oxygen to get in. Lastly, do you want to do a flow through, a static, this endpoint, or an intermittent flow? They all have their advantages and their applications. The only real advantage of an endpoint is that you only need one electrode and you can be incubating as many bottles as you want, as many organisms as you want. So if you have hundreds of larvae that you want to measure, you can have them incubating simultaneously and take one water sample at a time, and you're just looking at the change in oxygen from your beginning to your end. So there's just a point on either end, and you don't know what the oxygen is doing in the middle. But so if you need lots of data or have limited time or limited numbers of electrodes, that's the way to go. Flow-through is great for organisms that don't swim too much. If you have a change in the respiration rate, the calculation for, so basically you have an electrode on either end of a chamber and you're pumping water through it, and so you know what the oxygen going in and the oxygen coming out is, and the difference between those is your respiration rate. If the respiration rate is changing on some time scale, then the calculation for looking at the difference in those is not trivial to figure out what the metabolic rate is. This assumes a steady state situation, which is rarely the case in actively swimming animals. Closed or static system, you just put an animal in a chamber with one electrode in it and you watch the decline in oxygen over time. That's fine, except that while the oxygen is decreasing, the CO2 is increasing, and the oxygen may become a limiting factor at some point as well. So depending on the question you're asking. What I'm going to demonstrate today is what's called intermittent flow respirometry, and this is a compromise between these two methods. You have one electrode, but you've got water flowing through, and you can maintain the oxygen level of the water coming in, the CO2 level of the water coming in at any level you want, and then at intervals you switch the flow so that rather than going to some gas equilibration column, it's just circulating through the chamber. So the flow doesn't change in the chamber, but every so often you shut off the outside influence and the oxygen will decline, and then you turn it back on and it comes back up. And so you can maintain this level. You could do 20 experiments over an hour and just look at the oxygen change for a few minutes at a time, several times in a row. I don't know if I explained that well, but basically, imagine these axes, the y-axes are very different. You can maintain the oxygen over just a few micromoles here and allow it to consume for just a few minutes and get a rate. Whereas this one, you're usually putting them in a much larger chamber and requiring oxygen to drop over a much more gradual period of time. For example, if you have to allow your respiration rate to take 12 hours, you have to put it in a chamber that's big enough that it's not going to consume the oxygen down in the first hour and a half. In this situation, you can have them in a much smaller chamber, maintain the oxygen for as long as you want, and then after a 24-hour acclimation period, just flip a switch and it becomes a closed system for a few minutes. So it's much, without actually disturbing the organism at all, you can change it from an open system to a closed system. All right. So there's a very good book on measuring metabolic rates. It focuses mostly on terrestrial animals, but there's a good chapter on aquatic respiration, and a lot of the principles from the other chapters apply as well to aquatic systems. And this is a quote from his introduction that I thought was applicable. It says, "It's possible to measure metabolic rates without understanding what you're doing, and in doing so you may think or hope that the data you acquire are accurate. In fact, this approach is pretty much the rule, but my hope is that this text will discourage this approach. So it's much like CO2 chemistry. You can go out and measure alkalinity and pH and DIC using methods that are not in the best practices guide or with hands that are not as experienced as others, and you can hope your data are adequate, and in some cases they are, but it is not trivial, I guess is the message. All right, so I'm going to switch gears a little bit to talk about oxygen transport in the blood specifically. I thought I'd open it up for questions or discussion if anybody wanted to at this point, or if we're all happy to keep on chugging, I can do that too. How many people in here actually imagine that they might measure some physiological process in an organism as a function of CO2? Okay, good. It's more than I expected. So, great. How many of those are calcification? No? One or a couple? Okay. All right. Do you have a question?
I just wanted to add a comment. I was actually emailing Adam recently about optode versus electrode, and he's all about optode.
Yeah.
And I've actually done the Martian Manahan method with larvae, so I can tell you about how that works.
Mm-hmm.
But I haven't tried his microtiter plate method.
Okay, great. He taught me that method as well, and it seems to work well for large organisms. So when we go out to sea for extended periods of time, maybe 2 weeks, in order to get sufficient numbers to look at a size spectrum and at different temperatures and different oxygen levels, there's no way you'll get that much data in a 2-week cruise using individual electrodes in each sample, unless you've got an army of people and a whole lot of money to bring out as many oxygen meters and electrodes as you'll need. So the endpoint measurement works really well, but admittedly the precision is much, much lower than you might want if you're trying to tease apart some of these variables or look at really subtle changes in metabolism. If you're looking at the deep versus shallow that I told you about, that method works just fine. You've got the precision that you need to see that big of a difference, and you're seeing the body mass effects and the temperature effects, but you would be hard-pressed to see a change due to a subtle CO2 difference, perhaps. So, good point. Okay. So 2 particular cases where oxygen consumption would be expected to decrease as a function of CO2. One is metabolic suppression. So there are organisms out there living in intertidal zones, for example. Those are the most thoroughly studied in this respect, that as the tide goes out and they're left in a tide pool, the CO2 goes up, oxygen goes down, and they will shut down metabolism and wait for the tide to come back in. And in some cases, they'll be shut down for weeks or longer. And so it's a very effective metabolic suppression. They reduce energy production and consumption to just a fraction of what it is under normal conditions. And in some cases, there's good evidence to suggest that this is triggered by either CO2 or pH rather than the oxygen that is really the limiting factor. They use CO2 or pH of the cells to trigger the cascade of events that shuts down metabolism. And so in some organisms, especially those that naturally would undergo metabolic suppression, you might expect an increase in CO2 or decrease in pH to cause a reduction in metabolic rate. And that's been observed in certain organisms. And this doesn't mean that it's possible to tell that that's what's happening. If you just measure metabolic rate in 2 treatments, it may very well be that there's something else going on, as in that pie graph that I showed. And it's not possible to know. But if you have a reasonable expectation that your organism does shut down metabolism in response to intracellular pH, then you can demonstrate under an acidification scenario that actually happens and be comfortable with your interpretation of that result. The second is where oxygen transport limitation occurs due to pH sensitivity of respiratory proteins. So that acidification of the blood that drives oxygen off at the tissues where it's needed as I mentioned, can cause problems for oxygen uptake at the gills. And so any place where you are already pushing the limits of oxygen transport, if you have an animal with a very high metabolic rate, working at a very high temperature and swimming at a very high rate, then it's already going to be stressing the limits of its oxygen transport capacity. And if you add CO2 to that insult, then you might expect a decrease in capacity for oxygen consumption at that point. All right. So this is just a graph showing metabolic suppression in several common organisms that are capable of doing this. And the tolerance in terms of days of anoxia is correlated with the percent suppression of metabolism. So ATP as a percentage of the control condition, ATP turnover rather. So if you are producing ATP at a very small fraction of your control rate, you can survive for a much longer period of time in anoxia than you can if you are shutting down metabolism to 30% of your control level. So things like nautilus have been shown to shut down metabolism to about 30% of its normal aerobic rate. Some intertidal snails do better than that. Turtles can overwinter in anoxic ice-covered ponds. They shut down for extended periods of time. And the champion are brine shrimp. These are the sea monkeys you can order in the mail in their encysted state. So they can withstand nearly 4 years of complete anoxia because they shut down almost completely. There's hardly a measurable metabolic rate at all. Oxygen limitation due to transport stress is, I guess, difficult to demonstrate as well by measuring metabolic rate, but you can pinpoint animals that you expect to be more sensitive. There's going to be a difference in the CO2 and pH of blood that's near the gills, where you're getting rid of CO2 and taking on oxygen, versus at the tissues where you're taking up CO2 that has been produced by the cells. And that pH difference helps to drive oxygen off at the tissues and take oxygen up at the gills. The purpose of the respiratory proteins is to increase the total transport capacity of the blood in the first place. If you just have a partial pressure of oxygen equivalent between these two treatments, you've got oxygen dissolved in the seawater, same on both sides, so the partial pressure of oxygen in both of these chambers is the same. But if you add hemoglobin or hemocyanin that binds oxygen and pulls it out of solution, the total oxygen concentration is about 60-fold higher in this case on the oxyhemoglobin side. So you dramatically increase your oxygen carrying capacity. And so you can imagine that it's possible to actually take up oxygen from fairly hypoxic seawater, deliver it to the tissues, and by acidifying this solution, all of this oxygen is released and you suddenly have a very high partial pressure of oxygen at the tissues. So you can actually have a high partial pressure of oxygen higher at the tissues than it is in the environment. And this just shows pretty much the same thing, that as you increase the partial pressure of oxygen, the oxygen content of the blood increases as you would expect, but the vast majority of that is actually bound to the pigment. This line here shows the amount actually dissolved in the plasma. There's very little oxygen actually dissolved in plasma. Most of it is bound to respiratory pigments. Physiologists normally characterize the affinity for oxygen using a P50. That's the partial pressure of oxygen that gives you 50% saturation of the respiratory protein. So half of the binding sites are occupied by oxygen at this partial pressure of oxygen. So if you have a higher P50, so if your P50 is here, then this curve would effectively be shifted to the right. That means you have a lower affinity for oxygen. A higher P50 means a lower affinity. It requires more oxygen in the environment to saturate the blood. If the curve is shifted to the left, you've got a lower P50, which is a higher affinity for oxygen. So this is a curve for human blood again, and you can see that at the partial pressures that you find in the arteries, the blood is fully saturated. Under resting conditions, the partial pressure of oxygen drops to about 40 millimeters mercury, and you've only dropped a little more than 20% of the oxygen in the blood. There's still a huge reservoir of oxygen that is not tapped under resting conditions. When you start to exercise, the PO2 drops further, and you'll take advantage of maybe 60% of the oxygen carried in the blood. But you've still got some reserve. And when you're really stressed out, that's when you're producing the most CO2, producing the most protons that go into the blood, and you can actually take advantage of the last bit. I'll demonstrate that in a minute. So the Bohr effect is a measure of the sensitivity of oxygen binding to pH. All you're doing is measuring the P50 at different pH levels. Plotting log of P50 as a function of pH, and the slope of that relationship gives you the Bohr coefficient. So higher pH is giving you a higher affinity, which is a lower P50. So as you acidify the blood, the P50 increases, which means the affinity decreases. So if you have acidity at the level of the gills, you're shifting the curve in this direction, and it takes a higher partial pressure of oxygen in the environment to saturate your blood. At the tissues, it's an advantage. You shift it in this direction, and it releases more oxygen to the tissues where it's needed. But at the gills, it's a disadvantage. And these curves here show that once you've dropped to a low PO2 at the tissues, if you acidify, you've released a lot more of the oxygen bound in the blood than at these higher pH levels. There's also something called the root effect, and this is found in some fishes. And what this is a decrease in the total carrying capacity of the blood at a lower pH. So you're getting some Bohr effect, which is a right shift of this curve, but there's also a decrease in the total capacity to carry oxygen. That's called the root effect. And this is believed to assist in filling a swim bladder. So a buoyancy device in a fish, you can drive oxygen from the blood into this bladder and fill it up with oxygen gas against a very high partial pressure gradient by acidifying the blood near that swim bladder. So you can imagine in a fish that has a very pronounced root effect that the total oxygen carrying capacity is going to be reduced under an acidification scenario. And exactly how significant that is has not been looked at all, to my knowledge, in this context. This also brings home the message that there are a lot of complexities to this that I'm skipping over. For example, there are fish that have, I believe, 17 or 18 different isozymes of hemoglobin. So different proteins, each with their own unique affinity, that are produced in varying concentrations at different under different conditions. As far as I know, in the squids, there's only one type of hemocyanin, but that's probably just because nobody's looked in detail yet to figure out how many there actually are. So this story can get much more complicated than we currently know it to be. There's also a temperature effect to consider. As I mentioned, when an organism is in high temperature, it's got a higher metabolic rate. It's going to need more oxygen, but that's also when you have the lowest affinity for oxygen. So add a temperature insult to a CO2 insult when an animal is demanding the most oxygen, and that's when you're going to get into limitations of oxygen transport. So it's just sort of a ugly schematic showing how this is working. So you've got cells at the tissues producing carbon dioxide, diffuses into the blood, reacts with water, produces protons. Those protons bind to hemoglobin or hemocyanin, driving off the oxygen that then is free to diffuse into the tissues where it's needed. Imagine this red blood cell traveling through the body then to the gill, where you've got high oxygen concentrations diffusing in, shifting this equilibrium in the opposite direction, kicking off this proton, which drives CO2 production, and that CO2 can then excrete out. So most of the CO2 in the blood is carried in the form of bicarbonate, and once it gets to the gills, it's converted to CO2 that diffuses across gill membranes. So that's sort of an overly simplistic view of the whole system and how it's working. And that enzyme carbonic anhydrase that I mentioned is catalyzing this reaction. So you have high concentrations in the gills and in the tissues that facilitate this process. So imagine now that you have higher CO2 in the seawater in the gills than you normally would have, and this process of oxygen uptake by the blood is going to be inhibited. So some of these considerations and looking in detail at some species of squids led Hans Portner to suggest that hemocyanin function is going to underlie dependence of squid on low environmental CO2 concentrations. He measured the pH sensitivity of squids and found, let me back up for just a second. So this slope says minus 0.23. That's a typical Bohr coefficient for a lot of organisms. Squids are -1 or greater or less, lower than -1 in most cases. So very pH sensitive blood, which led Portner to suggest that they rely on low environmental concentrations of carbon dioxide. He also suggested that they are operating at about the limits of body size, at about as high a temperature as they could possibly do, and that they also require very high oxygen concentrations. In other words, they're maximized already to support their metabolic rate in terms of how much oxygen they can transport. And any stress to that is going to lead to difficulties. This is a picture of a deep-living squid called Galatuthus. It's commonly called a glass squid. It's very transparent. So we're looking directly through the mantle muscle, the body wall, and you're looking at the gills and the hearts. They have 3 hearts. And blue blood. So when you deprive them of oxygen, the blood turns transparent. And when you add oxygen, it turns a deep blue. So we're developing optical techniques to quantify this in some of these deep-living species and see if they are equally sensitive to CO2 as some of the shallow-living squids. But regardless, squids that have very high oxygen demand require a very fine-tuned blood transport capacity. But because they have hemocyanin, which is extracellular, and it's a very large molecule, there's a limit to how much hemocyanin they can have in their blood. Hemoglobin is intracellular, found in red blood cells. You can pack a lot of it in there without interrupting viscosity. So the total carrying capacity of squid blood tends to be lower than similar fishes, and as a result, squids are known to pull all of the oxygen out of the blood on every pass through the body. So there is no venous reserve. I talked about in mammals, at rest, there's about 80% oxygen reserve. In squids, there's no venous reserve even at rest. That's partly because they have jet propulsion, which is the reason they have such high oxygen demand compared to fishes. Jet propulsion is very inefficient. They are required to take water in, pass the gills, and eject it at a very high speed through a very small funnel opening to generate similar speeds to fishes, which have a broad caudal fin. So it's this efficiency that drives their metabolic rates to such high levels, because they're found in the same environment as some of these pelagic fishes, and so they're effectively competing with them, which led Ron O'Dore to suggest that squids consume twice as much oxygen to go half as fast as a similar-sized fish. So what I'm doing here is building a case for the reason why squids should be especially sensitive to high CO2, given their constraints on their blood oxygen binding, extremely high metabolic rates. These are squids compared to mammals, benthic organisms, and the vampire squid and jellyfish down here. So extremely high metabolic rates. This includes the market squid, Lullaginidae, the ones you would find in most restaurants, and the omostrephids, which are the more oceanic species. So this is that quote from Portner, a little out of order. Squids may operate at environmental limits of temperature, oxygen availability, and body size. And when Portner said this, he was talking about ilex, which is found off of our coast here. It's a squid about this long. It's living in North Atlantic temperatures, fairly cold, and oxygen in the Atlantic is fairly high. So this is an animal that he suggested was already stressed out, even though its environment is not as extreme as you can find in other places. So we wanted to study an animal that sort of defies all of those characteristics. This is Dicidicus gigas. You've probably seen it on the Discovery Channel. I don't know if you guys can see that. Is there a light switch to bring down? So these are squids feeding on a bait ball. Just to give you some idea of their activity levels and their size. They're very aggressive animals. Not as aggressive as the Discovery Channel would have you believe. There have not been any deaths due to squids ever. They do not eat people. They eat small fish and krill.
How long are those guys?
Maximum size, they are about 2 meters. These are probably about 1 meter that they're showing here. We've been diving with them at night. They don't bother us. As long as you have a flashlight on, they stay at the periphery. So all those Discovery Channel shows you see are when they've chummed the waters and gotten them very excited. They've got them pictures of them feeding on tuna, but it's half of a tuna. It's been chopped up and dumped in the water. I don't believe they actually attack tuna. I got an email from somebody at the Monterey Bay Aquarium. They'd put a tag on a great white shark they'd had in captivity, and they released. And about a year later, it showed up in the Gulf of California on the exact dive spot where we had been diving with squids earlier that year. And he was worried that the squids were going to hurt his shark. I'm like, you're watching too much TV. I think he's feeding on my squids. These are vertical migrators in the eastern tropical Pacific. So nighttime, they're mostly up near the surface with some stragglers at depth. Daytime, they migrate down to about 300 meters depth. This is data taken from tags that you can put on the animal that record temperature and depth over time. So this is the oxygen minimum zone in the eastern tropical Pacific where these animals are found, which also happens to be a CO2 maximum zone. So the axes again have dropped out, but this is in the eastern tropical Pacific as a function of depth. So within a few hundred meters of the surface, you're reaching very high partial pressures of CO2. This is an 800 isoline here, and down in the core, you're at 1100 parts per million. So 200 meters, 400 meters, you're already at 1100 parts per million. So these animals are migrating to a depth where they're experiencing very high CO2 and very low oxygen. And even at the surface, their depth, if I can back up for a minute, they're mostly at around 100 meters to 50 meters. They're not right at the surface. And in some places, already at 50 meters, you're at extremely low oxygen and high CO2 in the eastern Pacific. So they're definitely experiencing some what we imagined would be stressful conditions. So we designed a ginormous swim tunnel that we could take out to sea. This is on board ship on the New Horizon. So it's just a big holding tub with a working section inside and pipes going around and a bow thruster from a ship as the propeller driving a current through the system so you can get the animal to swim against a current and simultaneously measure metabolic rate and control the CO2 and oxygen level. That was the theory. Anyway, so these are chillers here to drive down the temperature in this much water, surface water coming in at about 28 degrees Celsius, and we're driving it down to 10 degrees that they would see at 300 meters. That would take pretty much all night or all day. Plop the squid in, try to measure it. Oops. And I don't know if you know this about squids, but they produce ink, and lots of it. And they do it repeatedly, and you can't stop them. And it's gooey and messy and ruins everything. So that's taken from a submersible at the Monterey Bay Aquarium Research Institute. Ink got literally all over the ship. This is my student Al Nyack with an ink handprint on his head. So this is a squid in the respiration chamber shortly after we put him in. And this is that same respiration chamber a few minutes later. So it wasn't pretty, but we did manage to, so we drained the tank, fill it back up, bring the temperature back down, put an animal back in, and we did get some decent measurements. Let me back up to this one. Sorry. This is a correlation between swimming activity and pressure inside the body of the animal. We put a pressure tag inside the animal. We were trying to calibrate the animal effectively so that you could eventually put a tag inside the animal that would record the pressure as it swims by jet propulsion, and you could correlate that with the oxygen consumed and the speed they were swimming. And so we're partway there, but it's going to take a lot more work to get fully there. Eventually you'd be able to release an animal. Almost out of time.
No,
Okay.
I'm just standing by for the lights.
Okay.
Sure.
Gotcha. Thank you. So eventually we'll be able to put a tag inside the animal, release it into the wild, and get field metabolic rates theoretically from that. So that's the idea, and we're close. But most of the data I'll show you today, we ended up using smaller animals that fit in these chambers. Same species, but juveniles that we could dip net over the side of the boat. This is a gas equilibration column, so we're bubbling with a given concentration of oxygen or CO2, pumping it through these chambers, measuring oxygen on an intermittent flow basis. That's Rui Rosa, who was a postdoc with me, and now he's in his home country of Portugal as faculty there. We also measured blood oxygen binding. As I mentioned, it changes from transparent to blue. So these are cuvettes with hemocyanin from Dosidicus in it. One of the advantages of these animals is that you can get gallons of blood out of them. It's very easy compared to the crabs we'll try to extract blood from later this afternoon. And those are different oxygen levels. So the one on the right is fully saturated, very dark, deep blue. And we confirmed in this species the pH sensitivity that Portner had measured in other species. Highly pH sensitive with a Bohr coefficient less than 1. So there's that. So we expect that CO2 would limit these animals, at least under some conditions of high activity, high temperature, or low oxygen. And so we put them in these chambers, and you could record the oxygen consumption rate, which was highly variable and somewhat cyclic. And when you would introduce high CO2, which in this case we used 1,000 parts per million PCO2 certified gas mixture, bubbling it through this equilibration column, you would, in most cases, see some sort of a dip in the oxygen consumption rate right at the introduction of the high CO2, and then a return to control levels. But you rarely saw these very active spikes that you see under the high CO2 conditions. And we could go back and forth between CO2 and normoxic water without alerting the animal or stressing the animal out. And so again, you'd see a spike right when we return to control, and then a dip when we go back to CO2. And notice that this is a fairly low metabolic rate. In this case, don't worry about the units, but keep 25 in mind. When you get up to animals that are swimming much more actively in the chambers, and we could correlate swimming just by videotaping these chambers in some cases, and you could see the mantle contractions. When they have very high metabolic rates, up around 60 to 70 in this case, introducing them to high CO2 has a much more dramatic effect. So we did this for many different individuals, and if you correlate the oxygen consumption rate under normoxic conditions, normal CO2 conditions, with the metabolic rate under 0.1% CO2 or 1,000 parts per million, you find that there's a one-to-one relationship until you get up to some high metabolic rate, and then it starts to plateau. So at very high control levels, they don't seem to be able to maintain that high level under high CO2 conditions. So anything on this side of this line indicates that it actually had a higher metabolic rate under high CO2 than under normal CO2, and that only happened in this one instance. All right. So at 20 degrees Celsius, that was the other thing I wanted to say about this. I knew there was something. So these higher metabolic rates at this end are driven by higher temperatures. So these rates at this end are mostly 10 degrees. These animals are 20, and then a few of these up here are 25 degrees Celsius. And when we look at the animals that were measured at 20 degrees or 25 degrees Celsius, there was a significant reduction in metabolic rate between high CO2 and normoxia. There was not a significant reduction at 10 degrees Celsius. So at the temperature they would experience at 300 meters where the CO2 is highest, they wouldn't experience any reduction in metabolism. However, the oxygen is so low there that metabolism is dramatically reduced anyway. So it's the hypoxia that's driving the low metabolic rate in the oxygen minimum zone that allows them to survive there overnight. CO2 has no effect in the oxygen minimum zone, but this indicates that there could be a dramatic effect at the surface as CO2 increases in the coming decades. We also were able to quantify activity levels based on the cycles of oxygen consumption rate, and the number of active cycles decreased dramatically under high CO2 as well. So that supports the idea that this is a reduction in total metabolism at high activity levels or high metabolic rates. So this was published this year, and we concluded that the synergistic effects of temperature, oxygen, and CO2 might have the effect of vertically compressing the habitable nighttime depth range of the species. In other words, at night, they typically come up toward the surface, but as CO2 increases, the ability to come all the way to the surface will be reduced. That's where their temperature is going to be highest, their metabolic rate is going to be highest, and where CO2 will have its maximum effect. So that'll serve to keep them down, but they have to come up at night to recover from their daytime in the low oxygen zone. So they have to come up above a certain depth and stay below a certain depth due to CO2, so that nighttime depth range is potentially going to be compressed as a result of these three variables. So, any questions on that part? I have one more. How am I doing for time?
About 10 minutes.
Okay. So I just wanted to say one more thing about this, the idea that you can put a number on a critical CO2 level for species. This is just one attempt. There have been several. This is a paper that came out this year, Brewer and Peltzer in Science, suggesting that You can calculate a respiration index based on the ratio of CO2 to oxygen that indicates a limit to aerobic metabolism. And what that paper ignores is what I've talked about in the first part of this talk, which is that organisms can control the oxygen inside their cells and inside their blood to a great extent. You can have a higher partial pressure of oxygen at the cells than you would find in the environment, and you can have a lower partial pressure of CO2 at the cells than you could find in the environment if organisms are adapted to control that. And so the bottom line is that using a respiration index based on environmental PCO2 and PO2 tells you nothing about what the CO2 and oxygen levels are inside the animal that they're having to deal with. So that's the main thing that they've ignored, and I'll just show. There's a problem with the thermodynamic assumption as well, because organisms are open systems and they're bringing energy in, so, but I won't get into that. This is data based on oxygen for crustaceans. It was mostly measured by Jim Childress at Santa Barbara. And what this shows is 4 different regions: California, Hawaii, Gulf of Mexico, and Antarctica, with very different oxygen profiles, and organisms from those regions having very different critical oxygen partial pressures evolved to those regions. So what this is telling you is that as you move to lower and lower oxygen levels, organisms have adapted their blood to have a higher affinity, they've adapted their gills to have a higher surface area, they've adapted their ventilation rates, they've adapted the pH sensitivity of respiratory proteins, and all of these things deliver effectively the same level of oxygen to these species in these regions. They all have the same metabolic rate, but different critical oxygen levels. So there's a strongly evolved ability to compensate for environmental variation that can't really be quantified across species easily. I think if you looked in one region and within a depth and within a taxon, you can identify a range of PCO2 that begins to stress organisms out. But it needs to be experimentally determined, and it needs to be determined on several species so that you can generalize it a little bit. But as a single number that you can apply to the entire ocean, there's zero hope of doing that, in my opinion. All right. I'm going to skip that. I'll stop there. Yes.
This may be an ignorant question, but is the rate of diffusion of salt gas across the cell membrane controlled by molecular diffusion? Does the membrane have an effect on the actual rate?
The membrane can have an effect on the rate. There are kinetic effects involved. So, for example, in the case of the blood, and effectively pulling oxygen from the environment across the gill membrane into the blood. Doing this by controlling the gradient is what it's doing. So by pulling oxygen out of solution, binding it to hemoglobin, you're keeping the oxygen partial pressure low and maintaining a gradient from the environment into the cell. So that's one mechanism. Beyond that, there are active transporters that can enhance the rate of diffusion as well. Certainly carbonic anhydrase is going to similarly shift the equilibrium so that the gradient is better or more favorable for CO2 excretion or bicarbonate uptake. Yes?
Another probably ignorant question. What role does osmotic pressure have in the ion exchange ability for?
I think that's going to play a role in the ionic balance versus acid-base balance that I mentioned. Certainly, invertebrates, of course, are isosmotic with seawater, and so if you're transporting a bunch of ions in, you have to maintain that isosmotic relationship. So there's going to be some limit to what ions you can bring in and how many of them. There's going to be a charge differential, osmotic differential, and an acid-base differential between intracellular and extracellular and environment that is going to have to be actively maintained if it's if they vary from isosmotic with seawater, they're going to have to actively maintain that, which fishes do. Fishes are hyposmotic relative to the marine environment. So that has to be actively maintained.
So there are regulation methods.
There are regulation methods, yes. Yes?
You kind of skipped over the brewer article thing. I'd like to see that explained. I mean, you kind of passed over it.
Yeah.
Took the time to go
Sure.
into
So the basic idea in the Brewer paper is that you've got this respiration equilibrium equation. So organic carbon, oxygen going to CO2. What they're assuming is, so if you put this into a sort of equilibrium like this, the ratio of substrate to product will determine the thermodynamic favorability of the reaction. In other words, if CO2 is very high, this reaction is less favorable. If oxygen is very low, this reaction is less favorable. And at some point, you've reached equilibrium where there's no net. The back reaction equals the forward reaction. And so there's no free energy gain from that. Free energy is zero, and you can't derive any work from this reaction if it's going just as fast backward as it is forward. And so they've simplified this into what they call the respiration index, which is just log of the PO2 to PCO2 ratio. So they've suggested that when this ratio is 1, log of that goes to 0, that's an absolute limit to aerobic respiration. You can't have aerobic respiration going when this back reaction happens as fast as the forward reaction, which is true, except that that's only happening in a test tube. Inside organisms, they're actively providing oxygen, they're actively providing organic carbon, and they're actively getting rid of CO2. So you can have much higher concentrations of oxygen at the cell than you do in the environment, and much lower concentrations of CO2 than you do in the environment. And so it doesn't have any effect. So an organism is an open system, whereas this assumes that this is a closed system. That's the basic problem with it. So for example, organisms living at hydrothermal vents are living at a respiration index less than 0, which this paper suggests is an absolute limit. And so there are organisms, you can point to examples of organisms that are living below the absolute limit, and there are certainly a lot of organisms living below the level that they suggested was limiting to most organisms. I think they said a respiration index less than 1 was limiting to most organisms, but organisms in oxygen minimum zones do just fine at a respiration index less than 1. So the two primary faulty assumptions are that respiration equation proceeds toward equilibrium as if it's a closed system, which it doesn't, and that gas partial pressure inside the cells resembles the gas partial pressures in the environment. There are other methods that people have used to try to pinpoint critical levels, just harvesting all the data you can and trying to control for taxonomic differences and some of those things, and indicating at what PO2 most organisms become oxygen limited, for example, or you could do the same thing with carbon dioxide. But again, there's enough regional variability and enough evolved differences in organisms that those are hard to do effectively as well, or usefully. I think that's... so they made some predictions, just to get back to this for a minute. They suggested in areas where oxygen is permanently high that CO2 would have no effect, which is not at all correct. You can show that, well, in the case of the squids, for example, CO2 has a very dramatic effect even when oxygen remains high. Trying to remember what I was going to say about this last point here. So I think that's just a faulty assumption as well, that even if you don't have any change in oxygen itself, just the rise in CO2 will necessarily increase dead zones. I think the vast majority of organisms, I think, are not going to be sensitive to the types of CO2 changes that we're going to see as a result of acidification. I think squids, for example, are an exception on one end of the spectrum, and there obviously have already been shown some cases where there are effects, but I think at least to the limits of our detection abilities, we're not going to see effects in most organisms. I think a doubling or tripling of CO2 is going to be washed out in the buffering capacity of the animals, and they're not going to see it. We're not going to see an effect. Yes?
What's the role of the carbonic anhydrase in helping them deal with high CO2? Because in phytoplankton, they have carbonic anhydrase to concentrate CO2 inside.
Right.
Here it seems like it's having the exact opposite role.
Carbonic anhydrase can be, there are many different isozymes of that as well that specialize in converting the reaction one way or the other. And at the gills, you're getting rid of CO2, and it's effectively reconverting the bicarbonate that's carried in the blood to carbon dioxide that can easily diffuse across the membranes. So if you have higher CO2, concentration outside, a greater concentration of carbonic anhydrase could theoretically enhance your ability to excrete across that gradient. It's one possibility.
I was wondering if it was to accelerate the conversion of CO2 to bicarbonate, which then could be excreted out.
I think it could work in either direction. At least at the gills, it's usually reconverting the CO2 that's carried as bicarbonate back to molecular CO2. But like I say, it's has different roles in different tissues, and probably has many more roles than we currently know. So, yes?
What's the definition of a dead zone, and do squids exist in the dead zone?
There is no hard and fast definition of a dead zone. That's one of the goals of this paper, was to try and say, okay, a dead zone is when the respiration index drops below 1. They're trying to put a hard number on it, when I don't think you can really put a hard number on it. And so, I mean, it's kind of one of those, you know it when you see it. When there's less organisms there, then oxygen is likely what's limiting the biomass of those organisms. And when oxygen drops completely to zero, the only thing living there are anaerobic organisms, which are few and far between. So no hard and fast definition. And yes, you do find squids. The Dicidicus that I showed is certainly in a region that is considered a very pronounced oxygen minimum zone. And it migrates into that layer. It doesn't live there permanently. It has to come out. The vampire squid lives permanently at depths that are very hypoxic. They don't qualify as a dead zone by most definitions. Off California, the oxygen drops to about 1/4 of a milliliter per liter at 600 meters, and vampire squids do just fine there, whereas most organisms don't. But it's definitely not a dead zone. There's still quite a bit of biomass at those depths. So no precise definitions.
So do you think they were describing a physical term rather than physiological organism?
Can you describe them in physical terms?
Yeah.
A dead zone?
The rule of phase, what is it?
They were trying to describe it using physical parameters to pinpoint where animal physiology was limited. And by ignoring how organisms evolve to those physical parameters, they, I think, just missed the mark. I don't think you can put a hard number on it, because there's going to be some ultimate limit to what physiology is capable of, but I don't know that anybody's ever identified it or that it applies to most organisms. Most organisms are living well above this ultimate limit to what oxygen transport is, and there's no way to really identify what that ultimate limit is. So I think there's room, for example, organisms living in the eastern tropical Pacific permanently at depth. there aren't many that live permanently at depth. The vast majority of organisms there are vertical migrators. They come out of the oxygen minimum zone during the nighttime. And so I think there you've started to approach what is some hard limit to animal physiology, but there are clearly exceptions. And this definition of that limit doesn't make any sense.
Yeah, I
To be-
think we're going
to
to
be
have
blunt.
that be the last question. We're out of time for this session. If you have some more questions, I guess you can save them for when we come to the lab portion of this
Sure.
topic this afternoon.
Thanks.
Thanks, Brad.
Thank you.
Thanks, Brad. Can I get your little talk on here?
Can I? Well, I guess I talked about most of it. It's not saved as a PDF.